Ladies and gentlemen, we are ready for our final session of the day, which is session 3, Navigating Global Innovation Pathways from Scientific Discovery to Clinical Study. The chair for this final session is Professor Hongfa Tse, and the moderator is Professor Xin-Ge Yu. To start with all the presentations, I'd first like to invite Professor Hongfa Tse, Chair Professor of Cardiovascular Medicine, William M.W. Mong, Professor in Cardiology at the University of Hong Kong, on stage. His topic is Cardiovascular Regenerative Medicine, Gene and Cell-Based Therapies Using Adult, Embryonic and Induced Pluripotent Stem Cells. Over to you, Professor Tse.
Thank you for the invitation. So I think we just change a little bit of the topic about some of this imaging. So talk about some potential innovative therapy. Hopefully we can deal with some of the patients you actually pick up by the MRI or some of this imaging. So obviously one of the objectives is actually for patients who actually had a disease or aging. So hopefully we're able to regenerating the organ and tissue. And obviously there are various ways we can using it. Either we can using cell as one of the platform, you can using a biomolecule and even some biomaterial as a combination to try to achieve our role of regenerative medicine. So, and I will show you why we need to do this, because as a cardiology, we are quite happy early on we see there's a reduce in mortality, but upcoming you see there's actually rising up. So the problem is actually we saved a lot of patients after the acute myocardial infarction. Eventually we translate them to heart failure. And we're just delaying the process, but we are not killing the process. And indeed, if you look at the epidemiology, so this is a recent data just published in BMJ, look at the mortality of people who actually suffer from stroke, acute coronary heart disease are actually reduced because we now have more effective treatment. Unfortunately, we just have the damaged heart. We just delay the process, and subsequently a lot of patients will die because of heart failure. That's why you see a U-shaped curve, and eventually the people have more people will die after the heart attack. And obviously, if you look at all this as advance of treatment in various therapy for heart disease, and most of them are palliative because they don't actually addressing the fundamental problem, you're losing the heart muscle. So obviously, people are interested to put in cell and different things. Hopefully, we can be generating the heart. But unfortunately, comparing to other organs like the bone marrow, the liver, the heart have very limiting regenerative capacity. So on its own, they are hardly able to regenerating by themselves. So they need to have something else. That's why people think about maybe using stem cell and all this therapy. Hopefully we can be generating the heart as a therapeutic approach. And this is kind of diagram to show people are working on. Obviously we can using different type of cell. Either we can using cardiac tissue to get cardiac cell, mesenchymal stem cell, various type of stem cell, and then hopefully we're injecting that back to the heart and then regenerating the muscle. Hopefully we're improving the heart function. So this is all very simple concept, but hopefully this is the promises what we aim to do. Things are not that simple. So we start from doing that almost 20 years ago, and we actually start from looking into injecting the bone marrow stem cell from the patient back to their own heart. And hopefully we'll see some improvement. At that time, there's some belief that the bone marrow stem cell will regenerate to muscle. But what we did is actually we'll get patients who actually have blockade in the artery, we cannot open up, and hopefully we're injecting the stem cell to improve angiogenesis. So indeed, we actually published this paper in Lancet that actually is the first human proof of principle data. If we're injecting the bone marrow cell, it's actually improving angiogenesis in a small cohort patient. And then eventually move on to do a clinical trial. So this is what we call phase 1-2 clinical trial, randomized patient with sham control. And then we're injecting saline versus injecting stem cell. And we're able to demonstrate they actually does improving some small amount of heart function and improving the symptom. So we actually proved the concept. The bone marrow stem cell does actually improving angiogenesis. But this is not the ultimate reason why we need to do this, because we hopefully will regenerate the muscle. So the next trial we actually put into a patient who actually have much sicker heart. So they don't actually have reduced blood flow, but also have a dead myocardium. And unfortunately, when we do this, we fail to show any benefit. So the bone marrow stem cell will help angiogenesis, but not able to regenerating muscle. So that actually doesn't fall short of our expectation in terms of treatment. That's why we think about along the last 20 years, what can we do for those patients? Can we find a new stem cell that is more effective. So we try different stem cell. People in the old day try a skeletal myoblast. We try the bone marrow stem cell, it doesn't work. And then eventually, now we move on to the mesenchymal stem cell and also some of these other pluripotent stem cell divided tissue cell. And the concept is obviously with the advances of using iPSL or using more pluripotent stem cell, maybe we can actually generating true cardiac muscle cell to injecting back to the patient. So that may be the ultimate goal to do this. Obviously, things are more complicated, but people are trying different cell therapy. And what we actually aim at is actually using mesenchymal stem cell. Why we focus on mesenchymal stem cell? Because obviously been proven in a lot of animal study, they are relatively safe and effective in at least some of these animal models. And again, in different human animal study, they're using different type of allogeneic, autologous. They show some benefit in terms of large animal study. In fact, if you look at some of this early trial, also in some patient with myocardial infarction, it's shown to be very safe. I think the most important part is, but they only have modest benefit. If you look at some of this latest trial, one of the unique thing about this interesting study is actually comparing different type of mesenchymal stem cell. So they actually try to using bone marrow versus umbilical cord. And in fact, in the cell culture system, they found out that even the same we call mesenchymal stem cell, they have different efficacy. You're comparing the bone marrow versus the umbilical cord. Umbilical cord mesenchymal stem cell are much more better in terms of parenchyma ability, less senescent when compared to bone marrow. And in fact, in this trial, they're injecting them using intravenously into patients with heart failure in this early trial. They did actually show some benefit. Again, the other trial also been using edible tissue, mesenchymal stem cell. We know that people claim that the edible tissue are easy to obtain. The problem is that edible tissue, a lot of time obtained from obese patients during liposuction. And those edible sites are very bad cell. In fact, they actually have very poor inflammatory. So in fact, if you're actually using some of this edible tissue, allogeneic mesenchymal stem cell, this is a poor analysis for 2 human trials. Again, they failed to demonstrate any benefit. If anything, they actually have some deteriorating effect. So again, if you look at a kind of summary for all this different type of mesenchymal stem cell therapy, the one who actually show benefit, it probably are those who are using umbilical cord, those who are using bone marrow and as well as edible tissue, they are not that effective. Interestingly, this is actually a publication recently published a few months ago, actually demonstrate if you're using the umbilical cord mesenchymal stem cell to do injection in patient after myocardial infarction, they did actually able to demonstrate a improvement in heart function and also reducing in cardiovascular outcome. So again, they proved the concept. Number one, the mesenchymal stem cell may be at least safe, may be effective, but obviously you need to choose the correct type of mesenchymal stem cell. Not every mesenchymal stem cell will do the same job. And this is actually evolving. What we're now working a lot on is actually thinking about maybe the iPS cell may be a unique source that can provide the mesenchymal stem cell with this development of this iPS cell. So indeed, actually, our lab been spending in the last 20 years to figure out what kind of cell are best in terms of doing cardiac regeneration. So we're using iPS cell divided cardiomyocyte, we're using iPS cell divided mesenchymal stem cell. Eventually, we settled that maybe we should go for using the iPS cell mesenchymal stem cell. Why we need to using iPS cell comparing to some of this tissue cell like umbilical cord, edible tissue, bone marrow? Because obviously, if you're not dividing from somatic tissue, number one, if you're using iPSC, they are much more easy to control in as an industry scale, manufacturer-wise, and also it can be off-the-shelf treatment, but also more consistent in batch-to-batch. And also, we're able to demonstrate that actually if we're using a iPS cell mesenchymal stem cell, we'll be able to provide more healthy cell than a sick patient. Like we try to get the same thing from a sick patient who actually had progeria, we failed to demonstrating a benefit of mesenchymal stem cell. So the origin of the iPS cell is also even important. And obviously, we move on to do this. Actually, we're able to generating functional mesenchymal stem cell from iPS cell or from ES cell, and able now to demonstrate they actually are able to regenerating vasculature. So the good thing is actually because we're using a single cone, we're not able to see any tumorigenicity, and also they're very safe. I think this is also one of the important beauty with the mesenchymal stem cell. And also we're demonstrating in animal model, they actually are much more effective than the bone marrow mesenchymal stem cell in terms of revascularization of the high limb, ischemia, and also have a better efficacy. Then we actually move on to do a head-to-head comparison with the iPSL mesenchymal stem cell versus the ESL-divided cardiomyocyte. People think are probably the best cell for doing cardiac regeneration. And interestingly, what in this we're comparing using human ESL-divided cardiomyocyte versus iPSL-divided mesenchymal stem cell. In fact, we actually see that the iPSL mesenchymal stem cell is actually better than the cardiomyocyte we're injecting. The problem is most of the cells we injected, most of them, they die. Even we give immunosuppression, whatever, most of them, they don't survive. And if anything, the mesenchymal stem cell give you more parenchyma effect and also more angiogenic effect. That's why they actually get, in this large animal model or pig, they actually get a better efficacy. And obviously, we also move on, do we need the cell, okay, because they don't survive? Can we just using exosome? So this is another study we just finished. We plan to submit is actually using the exosome, isolating from the iPSL mesenchymal stem cell. Again, in this small animal study, we also demonstrating that even using iPSL mesenchymal stem cell exosome, they also have improved heart function after myocardial infarction in this preclinical model. And obviously, people will think about why the mesenchymal stem cell is effective, because when we look at the differentiation, they're never able to get to the cardiomyocyte. But again, one of the most important issues about myocardial injury, after the injury, they will self-perpetuating. The inflammation, the cytokine, and all the things activating simultaneously. That's why even after the initial injury, the damage to the heart is propagating. That's why whether we can modulating the inflammation may be a very important role in terms of dealing with some of this heart injury. So indeed, if we look at the mechanism that we did a lot of studies, look at why the mesenchymal stem cell potentially being useful, it obviously have limiting ability for doing transdifferentiation. Some of the exosome we mentioned about, they have a lot of parenchyma effect, but more important, they have a very potent immune modulation as well as the other interesting people are talking about is mitochondrial transfer. Rejuvenating the mitochondria also potentially play an important role in terms of the efficacy of this mesenchymal stem cell. And indeed, we actually move on now from 20 years ago to try the bone marrow, all the way now to using the iPSL or pluripotent mesenchymal stem cell, and eventually we decided we need to build a lab to manufacture those cells for therapeutic use in Hong Kong. So again, we now focus a lot on using different type of pluripotent stem cell, and hopefully we're able to generating a universal kind of therapeutic that we can actually using, and hopefully we can put up to the phase 1 and phase 2 clinical trial. And obviously, we need to follow up a lot of the manufacture process. And I think if you look at the procedure, when we're doing a clinical grade mesenchymal stem cell, and you need much more, we're doing a GMP grade in terms of the QC and also the manufacture and also the checking. And that's why you're able to do that. Hopefully, we're able to do a universal mesenchymal stem cell for different therapeutic. The other beauty about the mesenchymal stem cell is not only for heart regeneration, they now have been a lot of application, like for example in stroke that we're talking about today, other disease like eye disease, COVID, lung disease, inflammation, cystic fibrosis, they all now have potential in application in that. So hopefully, even we may or may not successful in heart regeneration, but the cell may have other potential application in the future. And again, we need to make it in industrial grade, so think about using that manufacture also not only the cell, but also the side product from the cell is actually the exosome. Can be also manufactured as a kind of GMP compliant product for application. In fact, if you look at this latest now, so actually there's some company already marketing some of the cell using iPSL divided mesenchymal stem cell being launched for using for GVHD, which is a rejection disease in patients with bone marrow cell transplantation. But again, if you look at a lot of other trials been also ongoing with using iPSL-based therapeutic, not only for mesenchymal stem cell, retinal disease, and also some work on cardiomyocyte are also ongoing. So if you look at the company in 2024, now we're working on iPSL. It's a huge number. And in 2 years' time, even more, because the iPSL will become off-patent. So everyone will now actually jump into the iPSL market because now the cell line will be off-patent. We can actually generating iPSL for different purpose and application. And obviously, you see that if you look at the number of the trial and also the product and also the publication, you see this is a huge number of the increase over the year in terms of the iPS cell-based therapeutic being developed over the year. And you can see that in various applications, not only for the heart, but also for brain and other things. So again, the other thing we also focus on, you see that even for iPS cell, mesenchymal stem cell, there are now a lot of companies already focused on doing different development. As I mentioned, GVHD, even kidney transplantation, and also COVID has also been focused on doing some of those. And again, this is one of the early approved products. This is a paper they published in Nature Medicine to using iPS cell, mesenchymal stem cell for treatment for GVHD disease. And the other thing we're working in Science Park, we actually had this stem cell lab for using what we call extended pluripotent stem cell, which is even more naive than iPS cell. One of the advantages of that, you can actually generating from iPS cell or from the embryo, but more so they actually will have been more immune privilege. And again, they've been, by Pental Leaf Group, they've been demonstrated they can generating EPSC for different species. And more important, we're also able to generating the EPSC to mesenchymal stem cell. And interestingly, as I mentioned, even mesenchymal stem cell, they look similarly. They will have a different property in terms of immunosuppression. If we look at the transcriptome RNA-seq, we can look at different type of mesenchymal stem cell. Again, although some of them are very similar, but a whole lot of them are very different, so that we think that maybe they will actually providing more efficacy. So in fact, when we're comparing the immunosuppression efficacy, the iPS cell mesenchymal stem cell, if anything, is better than the umbilical cord mesenchymal stem cell. And also in terms of kidney regeneration, this is a SLE model for immunosuppression. They also are more effectively reducing inflammation in the kidney injury compared to umbilical cord. And also we are now demonstrating that in a highly ischemic model, they are also more effective in regenerating the blood vessel comparing to that. And obviously, there are different cytokine profile we're working on, why they are better. But eventually, you can see that we're evolving to develop different type of mesenchymal stem cell. Hopefully, they will tailor-make for different disease and application. And hopefully, eventually, we're able to tailor each of the disease for different type of mesenchymal stem cell. So those are the clinical trial now, I think will be upcoming in our unit. So we are now planning up a mesenchymal stem cell trial for heart disease. We're also planning up a iPS cell divided mesenchymal stem cell for actually treating patients with heart disease. And also we are also dividing our EPSC mesenchymal stem cell. Hopefully we can actually go to the clinical trial very soon. And this is one of the projects we're supporting by the Hong Kong government. So we actually planning to work on to generating GMP-grade iPS cell mesenchymal stem cell for patients with heart failure. We're also working on other company to look at using mesenchymal stem cell for treatment of stroke, and also some other Japanese company to dividing EPSC divided trophoblast and exosome for treatment of vascular disease. Finally, some of these cells need maybe actually other element. So in another delivery. So also we work on other with InnoCenter to look at how we can actually packaging the cell more effectively. So like some of the microgel, using different energy for delivery, can be encapsulating the cell to make it more effective. So those are the work we are also working into that. And so if the time allows, next time I will show you some other data we're working on that. So in conclusion, so regenerative medicine is an innovative and fast-moving field that applies to restore function in cell and tissue. And now we're actually into the phase that hopefully in Hong Kong and also in the Science Park, able to get to this manufacturer for this product based on this ATP criteria using with the development of GMP lab. And more important, some of these products, hopefully we're able to get into the clinical trial for different applications very soon. Thank you for your attention.
Professor Deng, would you please remain on stage? Now I'd like to invite Professor Xing-Ge Yu to present a souvenir on stage as a token of appreciation. Thank you very much for the presentation.